I'm John Housley. I'm a group leader at the Babraham Institute near Cambridge. So I run a research lab in our epigenetics program, and I'm also head of our knowledge exchange and commercialization team. The Babraham Institute is a life sciences research institute. We have a particular focus on aging, so why organisms age and why aging is associated with increased risk of disease and physiological decline. Our long-term aim is to improve health span. So that's the amount of time that we spend in our lives free of severe disease and free of age-related decline. So my lab works on the way in which cells respond to their environment. So we've done a lot of work on how they adapt to environmental change, but what we're becoming more and more interested in is the way in which they react to changes that come about during aging. So as cells age, they undergo all sorts of changes. These are referred to as the hallmarks of aging, and basically can be summed up as everything goes wrong basically at once. The problem is that we don't really understand why. We know that there must be some sort of driver that leads into this cellular aging process, but we don't know what that is. So what we're trying to do is work that out in yeast. And although yeast are very different from us, yeast cells are also eukaryotic cells, and they share a lot of our physiology and a lot of metabolism with human cells. So if we can learn what drives aging in yeast cells, we think we could then translate that forward to human cells. So our recent research addresses the effect of diet on the aging process. So we've known for upwards of a century that organisms that consume very few calories live longer and healthier lives. The problem is that the number of calories that you have to consume to actually achieve this effect are borderline starvation. So even if we can spend our lives consuming the right amount of calories to achieve this effect, it's incredibly unpleasant, and so no one really does it. So what we asked in this research is whether we could achieve similar effects by just changing the diet. So what we did was to take budding yeast, which are a classic model organism for aging, raise them on different diets. And what we found is that as we changed the diet away from rich glucose, which is what they're normally raised on, we found that although the cells didn't really live any longer, they didn't show the same markers of aging pathology and loss of fitness that we see on their normal diet. The next steps for this research are to take what we've learned about the way the metabolism is different under these different diets and see if we can re-engineer yeast to show the same increased health span but on a normal diet. And what this will mean for people is that this will potentially reveal pharmaceutical targets that we can use to improve health span on normal diets.